One last thing. So we're looking at the sequencing that BioMFX is doing, tell us a little bit about the methodology of really the collection and what sets us apart.
Yeah. And let me use an analogy for that, right? Because I'm guessing the vast majority of people that are watching this are not bioinformatics sequencing experts. And so this is all a learning process for everybody, including myself. So it's a distinction between these two types of sequencing technologies. One is called 16S ribosomal RNA sequencing, and the other is called whole genome sequencing. The analogy I usually use to explain the two is 16S ribosomal RNA sequencing is what the vast majority of stool tests use. It's technology that was developed decades ago. It's old technology, and basically published studies, the American Gut Project, all of the big microbiome research institutes have come out and said that you cannot use that technology to identify bacteria accurately to the species level. It's limited in how it is, because what you're doing is you're looking for the 6, 7, 8 different variable regions of the bacteria's DNA. And if you find them, if you find 2, 3, or 4 of them, you might put them together and go, okay, this is likely this bacteria. The analogy there is if you take an individual, right? Let's say we take you, Trish, and we're trying to identify you through pictures. And we've got a picture of your elbow, we've got a picture of your tip of your nose, a picture of your shoulder, the back of your head. So we've got 6, 7 pictures of different parts of your body. And we look at those different pictures and we're trying to figure out who that is. Now, we might be able to identify some things just from a picture of your skin. We'll say, okay, this is not an African-American person. So we've narrowed that down. This is a person with dark hair from the picture on the back of your head. From the picture of your nose, we might be able to tell that's a female, right? So we can use it to narrow down, but can we tell that that's Trish when we're picking you out of 1,000 people, right? We probably can't. So if you have 1,000 people, you've got these 6 pictures, you might be able to narrow down the 1,000 people down to maybe 100 who fit some of those descriptions, but you cannot tell accurately which one of those 100 people that is, right? So you make a guess and you go, more than likely it is this person. That's basically what they're doing. So when you see the presence of this bacteria, this bacteria in those tests, they're making a guess as to whether or not it's those bacteria. It's not absolute. And that's the thing that they don't tell you about the test. Now, the new technology is called whole genome sequencing. Whole genome sequencing means in order to identify a bacteria, we have to look at their entire DNA sequence. We have to find the entire DNA sequence of that bacteria before we identify it as that bacteria. Now, the analogy there is instead of that picture of 6 different parts of your body to try to identify you, it's a clear, high-definition resolution picture of you. And that's easy to look at that and go, that's Trish, and that eliminates all the other 999 people in the crowd, right? That's the analogy. So we've gone from that kind of primitive sequencing technology, which now is very, very cheap to run, and is probably the main reason why most of these companies still use it, because you can run it for, less than $20, $30 a sample, versus whole genome sequencing, which is still more expensive because the technology is newer. Requires a lot more to do it.
Fantastic. Wow. Thank you for that. That was wonderful. I appreciate that. And we're going to go more in depth with this in our future videos as we go step by step, section by section with the test with Quran. But thank you so much for explaining why we got into this. I'm super excited, like I said, to take the test and expose this to all of the people that follow you and support Microbiome Labs. So stay tuned, everybody, and join us on the next video. Thanks so much.
Thank you.